The Lyapunov exponent estimates whether two neurons respond in the same way to the stimulus or whether they start diverging from each other as the stimulus is repeated. And to estimate that, it's the log of the response to the stimulus, measured by the slope of the PRC here, times the probability distribution, which is the steady state distribution of the phases lambda phi, calculated from the stochastic map shown in the previous figure. In C, you can see the Lyapunov exponent calculated analytically from the PRCs and the noise terms, and the noise seems to smear out the Lyapunov exponent and the fractal behaviors don't go away. So it seems as though even in a noisy condition we still have positive Lyapunov exponents. In D, we actually started simulating networks of 100 cells, and they all receive common noise input as well as independent noise input. And every time the network is stimulated, we update the phases of the neurons according to the phase of the neuron at the time of the stimulus and the phase response curve. We ran 3 network simulations, one where the networks of neurons are receiving no stimulus and they slowly synchronize. And then we also ran simulations where we're stimulating at frequencies that cause them to synchronize, shown here in F, and at frequencies that cause them to desynchronize because the response to the neuronal network has a positive Lyapunov exponent, as shown in E. Here are 3 examples of network simulations that we ran. All of them have 100 neurons, each which receives common noise input and some independent noise to each neuron. The common noise is enough to cause all the neurons to fire. In the left panel here, we are seeing the neurons rotating in phase. So every time they cross this line here, the neurons are firing at action potential. And the common noise is causing them to synchronize up. Below you see a histogram of the neurons as a function of phase, so you can see the phases clustering up. This represents the 0 stimulus amplitude line on this graph below. The graph in the middle shows what happens when we start stimulating neurons at about the same frequency that the neurons are firing. So every time it fires, you'll see a little yellow flash of the screen. This stimulus. Finally, in panel G, we decided to add noise to the stimulus period. And this is measured in coefficients of variation. So 0 means periodically stimulated, and 0.2 means a 20% standard deviation in the interspike interval period. And as we increase this noise in the period, you see that the synchronizability of the network, or measured, increases, so the efficacy of the deep brain stimulation for desynchronizing the network decreases. This is consistent with the findings of Alan Dorval's work, but would be explained by a different method than he explained it. In figure 4, we looked at how deep brain stimulation may excite axons that are synapsing onto neurons in the striatum and the subthalamic nucleus. And we measure both the phase response curve, as you can see in C, of the neurons due to excitatory inputs, and immediately below it for inhibitory inputs. And then we combine those two together to have the effect of both the excitatory and inhibitory synaptic inputs into the neuron, as seen in the panel on the right in C. And then we measured the Lyapunov exponent as we varied the stimulus amplitude and frequency over a wide range, and saw similar graphs. What we found is that there continued to be a region that had a positive Lyapunov exponent, indicating that even if the stimulus was not directly affecting the neurons, but exciting both excitatory and inhibitory synaptic inputs into the basal ganglia that we could still elicit a chaotic behavior. In E, we're looking at what the stable, steady-state distribution of neurons are with respect to the stimulus, 0 being this time of one stimulus, 10 being the time of the next stimulus. And you can see at one stimulus amplitude, stim 0.45, we get a distribution that looks very similar that observed in the Hashimoto paper. We'd like to thank NIH, NSF, and University of Minnesota's Grant and Aid program for the support for this research. You may contact me at tnetoff@umn.edu if you have any other questions or comments. Of course, you may go to the paper for the much more detailed description. You also may visit my website, neuralnetoff.umn.edu, where we have a lot more information about this and other publications relating to this. We realized in John Milton's book in chapter 8 with John Hunter, titled Controlling Neural Synchrony with Periodic and Aperiodic Stimuli, they go very nicely into detail about how periodic stimulation can result in chaotic behavior and desynchronization of populations. And they point that this could also be used for treating epilepsy or Parkinson's. Another paper that's about to come out, it's in the archives, is by Eric Shea Brown, where they do very similar work, much more analytical detail, but it's of a bursting neuron and showing that at certain periodic stimuli they can get chaotic behavior with the bursting neuron. So we just wanted to cite these papers as an epilogue to this paper.